So good morning, everyone. Welcome to day 2. My name is Maria Squire. I am an associate professor of biology at the University of Scranton, and we are going to be covering structure and function of the respiratory system today. I have a handout for you. There's 2 for your table. There you go. 2, 1, and 1 for each of you. And there's 1 for you.
Thank you.
You're welcome. 1 for you. Sure. There's 1 for you. 1 for you. Here's 1 for each of you. Here's 1 for you. Do you want one for your member who's displaced?
Sure, yeah. Well, she's not, well, she meant Tara, but.
Yeah. There you go.
Thank you. For him.
Oh, okay. So does everyone have a handout? Yes? Okay. I want to introduce myself, tell you a little bit about me before we get started. I attended Freedom High School, which is in Bethlehem, Pennsylvania. It's about an hour and 15 minutes from here. And then I got my undergraduate degree at the University of Scranton. I majored in biology and Spanish. After graduation, I spent a year working as a laboratory technician at the University of Pennsylvania. I worked in the orthopedic research lab. I worked with shoulder and elbow mechanics, learned a lot of really cool things. Took some math classes that I needed as prerequisites for graduate school, and then I went to Stony Brook University, which is on Long Island. I got my master's and PhD in biomedical engineering. When I was nearing the finish of my dissertation, I applied for faculty positions and landed a job at the University of Scranton in the biology department, and I've been there ever since. In August, I will be starting my 9th year there. It's really hard to believe that I've been there for that long as a faculty member. The courses that I teach at the university, I teach anatomy and physiology. It's a course for first-year students majoring in exercise science, nursing, occupational therapy, and community health education. I also teach a sophomore-level general physiology course, which is taken by exercise science majors and also our biology majors, most of whom are in the pre-med track. And then I teach a skeletal biology course every other spring. This is a course in my specialty area. So my research interests focus on bone and bone morphology. I look at the structure of bone, and I look at how the structure of bone changes with changes in factors like weight bearing and hormones. So my dissertation work was sponsored by a NASA grant to look at the effects of weightlessness on bone, and now I'm looking at annual changes in bone in birds. So that's a little bit about me. Our task for today is to cover anatomy and physiology of the respiratory systems with a strong structure-function approach. I gave you the objective at the top of your handout. We are going to do some comparative examination of the respiratory system in vertebrates, so across vertebrates, talking a little bit about adaptations across vertebrates. And then we're going to look at structure-function relationships at a number of levels, so organism, organ system, tissue, cell. I will try to compete with the lawnmower. If you can't hear me, just let me know and I can talk a little bit louder. I just wanted to start with a couple of key terms that we're going to need in the beginning of our discussion of the respiratory system structures. We need to talk about the respiratory medium. That's going to be the source of oxygen. And so we have 2 different respiratory mediums that we're going to discuss depending upon the organism. So we have air and we have water. If we look at the amount of oxygen in these 2 sources, in these 2 respiratory mediums, per unit volume, air has significantly more oxygen than water does. And when we look at water itself, the warmer the water, the lower the oxygen concentration by volume. Also, the saltier the water, the lower the oxygen concentration by volume. And so this presents some unique challenges for aquatic animals as compared to terrestrial animals. And we'll talk about how the aquatic animals work to overcome this challenge. Can everyone see that okay?
I can see through that one, it's not mine.
Okay. We also are going to talk a little bit about how the respiratory surfaces are designed. The respiratory surface is the structure where the exchange of gases occurs. And in order to talk a little bit about the respiratory surface, it's important to do a little bit of a review of Fick's law of diffusion. So I'm going to define each one of these terms for you, and I'm going to highlight the ones that are most important for our discussion of the respiratory surface today. And so Q is the diffusion rate. You didn't think you signed up for a math class at 8 o'clock in the morning, huh? These equations are just really important to understand why these surfaces are designed the way that they are, so structure-function relationship. So Q is the diffusion rate. Delta C is the concentration gradient. Concentration gradient is in the numerator, so the bigger the concentration gradient, the faster the rate of diffusion. A is the surface area. Again, it's in the numerator, so if we increase the surface area across which diffusion takes place, the rate of diffusion will increase. Here's a handout for you. Beta is the lipid solubility. Of the substance that is diffusing. This is important when we talk about diffusion across cells, since the cells have a lipid bilayer for the plasma membrane. If it's lipid soluble, it will diffuse more easily. So those are all of the terms in the numerator. In the denominator is the square root of the molecular weight. The higher the molecular weight, the slower diffusion is going to happen. And then delta X is the thickness of the surface. The thicker the diffusion distance, the slower diffusion is going to happen. Okay. So when we talk about the respiratory surface, surface area and thickness are going to be really important. So the respiratory surfaces. Is there a question?
I didn't know what it was.
Oh, molecular weight. I'm sorry.
Just a lot to write down real quick.
8 o'clock. Thank you.
Yep. Hit the wrong button on my slide advancer here. So when we're talking about respiratory surfaces, they're going to be designed in a way that maximizes the surface area and minimizes the thickness of the surface. And what I often discuss with my students is if there were to be double doors here and I open just one of them, they would be able to exit the room at a certain rate. But if I were to open both of them, they'd be able to get out faster. So if you increase the surface area, diffusion can happen more quickly. Likewise, with the thickness of the surface, we talk about how long it takes to walk a certain distance versus double that distance. And that sort of helps them to see that the thicker the surface, the longer it's going to take for diffusion to happen. Okay. Is that idea okay with everyone? Yes? Okay.
What is the definition of diffusion before we move on?
Sure.
What are we diffusing?
Sure. So diffusion is the movement of a substance from a region where its concentration is higher to a region where its concentration is lower. And diffusion is a passive process, so no energy needs to be expended for diffusion to happen. Okay, so one example of diffusion, in the morning you make a pot of coffee, the concentration of the aroma is high around the coffee pot, and gradually the people upstairs start to smell it because the particles move from where the concentration is high to where the concentration is low.
Gotcha.
So that's diffusion. And what we're talking about here with respiratory system is the diffusion of oxygen and carbon dioxide across the respiratory surface. Okay? Good question. Everyone else good? Okay. So there's two respiratory surfaces that we're going to talk about. The first is found in aquatic animals, and the respiratory surface being the gills. So if we look at this image of a fish that I am using from the general biology textbook that we use for our students, you can see the gills here. So the gills are the respiratory surface. These are outfoldings. And you can see here that there are these little gill filaments. These little gill filaments create a large surface area. Another important feature of these gills is that they are in aquatic animals in water, so the surface is moist. There you go. There you go. But as I mentioned earlier, these aquatic animals have a challenge in that they have to overcome the low oxygen levels that are present in water. Okay? So they carry out a process called ventilation, which increases water flow over the gills in order to keep the oxygen levels up. And they do this using a process called countercurrent exchange. Okay? And here is a figure that shows what countercurrent exchange is, and I'm going to walk you through what it is and then why it's beneficial for maximizing gas diffusion. So there's two arrows here. The blue arrow is showing the direction of flow of water over the gill structure. The red arrow is showing the direction of blood flow through the gill structure in a capillary. Notice that they're going in opposite directions, countercurrent. This movement in opposite directions maximizes that concentration gradient so that it's always favoring the diffusion of oxygen from the water into the capillary. Okay? So, as the water is flowing over the gill structure, the concentration of oxygen is going to be maximum just as it's going over the gill structure. Here. As the blood is flowing through the gill, the maximum oxygen is going to be here, but notice that we still have a concentration gradient that favors the diffusion of oxygen from the water into the blood. And that's really important, is to always have that gradient. If you can imagine this red arrow going instead in this direction, here we would only have 15% oxygen in the water, 90% oxygen in the blood, and we wouldn't have a diffusion gradient anymore favoring the movement of oxygen from the water into the blood. So by having this countercurrent flow, there's always a concentration gradient favoring the movement of oxygen from the water into the blood. Is everyone okay with that?
Just for scale purposes, that's only like 5 millimeters probably in diameter, or the most of a typical fish. Like
Size? I wouldn't even think it would.
a millimeter?
Yeah, it's probably smaller than that. Yeah, it's probably smaller than that. I'm trying to think. I actually held a fish that we had caught in a pond a few weeks ago. Those gills are really tiny. I would guess it's no more than a millimeter. Yeah, yeah. I mean, certainly the size of the fish varies, and so you have a really big fish, and you probably have a slightly thicker gill. Are we good with that? Yeah? Okay. So that was all that I wanted to say about aquatic animals and gills. The next respiratory surface that I want to focus on is lungs. And terrestrial vertebrates use lungs. Some also use the skin as a respiratory surface, but for our purposes, we're going to focus on lungs. So frogs, birds, and mammals all use lungs as their respiratory surface. I'm going to get rid of this. And what is really important to keep in mind when we're talking about lungs is that the lungs are not in direct contact with the respiratory medium, right? So the oxygen is in the outside environment, your lungs are within. So there are conducting zone structures that we need to talk about, and these are the structures that get the air from the external environment into the lungs, okay? And then we have the lungs that we're going to talk about, and it's really important to keep in mind that terrestrial animals that have lungs, adaptations have been made to the lungs based on the size of the animal, also based on the complexity of the metabolic demands of that animal. So in some of the other biology sessions that you're going to have this week, you're going to be talking about cellular respiration. And so the rate at which cells carry out cellular respiration dictates how much oxygen they need, and so the metabolic demands are going to influence the size of the respiratory system structures. Does that make sense? Okay. So I want to talk about two different terrestrial animals, breathing in birds versus breathing in mammals. We'll start out with birds, and contrary to what you might think, breathing in birds is actually much more complex than it is in mammals. So these are the structures of importance when we're talking about respiration and ventilation in birds. The air enters the bird through the nostrils, passes through the pharynx, the trachea, and the bronchi. Birds also have these structures called air sacs, and so you can see there's a group here that are the anterior air sacs and the posterior air sacs. There's generally about 9 air sacs total, and they're grouped into those that are more anteriorly located versus those that are more posteriorly located. These air sacs do not serve as gas exchange, God bless you, structures. Instead, they are holding areas for the air. The gas exchange instead happens in lungs, and more specifically in structures within the lungs that are called parabronchi. Okay. So the air flows into the nose, down the pharynx, the trachea, into some of these air sacs for holding, then into the lungs where gas exchange happens. Then the old air moves into these anterior air sacs before leaving the bird's body. Okay. And it takes 2 ventilation cycles for the air to make its way through a bird. So this is an image that I am borrowing from an ornithology textbook that I had in my office. And these 4 pictures show the progression of the air through the bird. So here, the air is entering. It's in the bronchus here. With the first round of inhalation, the air enters into the posterior air sac. Okay? And then with exhalation, the air is pushed into the lungs. Okay. With the next inhalation, the air is pushed into the anterior air sac. And then finally, with the second exhalation, the air is pushed out of the bird. So it takes 2 full cycles for air to make its way through the bird.
Or is the air coming in and out?
Yeah, the picture is, it should be coming in and out the nostrils. Yeah, like this image was showing. This image makes it look like it's coming in mid-belly, like through the belly button or something.
That's why I was curious.
Yeah. The picture is not the best as far as showing accurately where it's coming into the body, but it was a nice figure in the sense that it showed the progression of air through two complete ventilation cycles. Okay. Funny that nobody yesterday took issue with that. What is unique in birds as compared to mammals is that with each breath, the oxygen is completely renewed, and so this maximizes the oxygen concentration in the lungs. There's no mixing of new air and old air, and so this gives birds an advantage over mammals at higher elevations where oxygen concentrations in the air are lower. Because they're maximizing the concentration of oxygen in their lungs, they're better able to perform at higher altitudes compared to mammals.
How do they increase concentration?
They're maximizing the oxygen concentration because with each breath, new air is coming in and it's not mixing with old air. So the air is completely renewed each time. So you're not in the certain confined volume of space, you have new air all the time. So you're not mixing oxygenated air with deoxygenated air.
You can't really do that. You can't eliminate old air.
No, you never exhale all of the air in your lungs. You always have what's called a residual volume of air in your lungs. So there's always mixing of old and new. Mm-hmm. We good with that? Okay. So now I want to talk about breathing in mammals. And breathing in mammals is most often studied in humans, so we're going to use humans as our model. That's also going to be the case, God bless you, when we get to the circulatory system later in the week. Humans are most studied, and so that'll be our subject there as well. When we look at breathing in mammals, again, the lungs are internal to the body, the air is in the external environment, so it has to get into the lungs somehow. The structures that get the air into the lungs are part of the conducting zone. Where the exchange of gases happens is part of the respiratory zone. And you all have a handout. If you want to turn to page 2 of that handout, the first item on the page I've made a diagram tracing the pathway of airflow from the nose into the lungs in a mammal. And we can work on filling that in as we go through this figure. So air enters through the nose, passes through the nasal cavity, and then into the pharynx. I've also given you a word bank on the side of the terms that you're going to fill in. So if you don't want to write the whole thing, maybe you just want to write a number next to that term and then just fill in the box with the number. Air passes from the nasal cavity into the pharynx, from the pharynx into the larynx, and you can see the larynx up here on this figure. Okay. From the larynx, air passes into the trachea. The trachea then branches into 2 bronchi. These are called primary bronchi. Each primary bronchus delivers air into a lung. So you have 2 lungs, right and left. You have 2 primary bronchi. These primary bronchi branch into secondary bronchi. Your right lung has 3 lobes. So So there are 3 secondary bronchi. Each one delivers air to a lobe of your right lung. The left lung has 2 lobes. So there's 2 secondary bronchi in the left lung. These secondary bronchi then branch into tertiary bronchi. Each tertiary bronchus brings air into a segment of a lobe of the lung called a bronchopulmonary segment. So each tertiary bronchus brings air into a bronchopulmonary segment, which is not a choice. And then these tertiary bronchi branch into bronchioles, which is a choice. And the smallest of the bronchioles in the conducting zone are called terminal bronchioles, which is also a choice. Tertiary bronchi, bronchioles, terminal bronchioles. There are a few more spaces. Those structures are part of the conducting zone. The last 3 structures are part of the respiratory zone. Okay? Is everyone good with conducting zone structures before I go to respiratory zone? Yeah? Okay. Following terminal bronchioles, these branch into smaller bronchioles called the respiratory bronchioles. The respiratory bronchioles branch into alveolar ducts, which end in these alveoli. If you look at these alveoli, they sort of look like grapes. If you have a bunch of grapes on a stem, the stem is like the alveolar duct, right? All of the grapes that share that common stem would form what's known as an alveolar sac. Okay? So the duct is the passageway through which the air flows. The alveolus is this cup-shaped outpouching like a grape. And then all of the alveoli that share a common duct form a sac, like a bunch of grapes. Does that make sense? So I brought a lung model, and I'm going to walk around with it so that I can show you what some of the structures are. This is not showing the nasal cavity, the pharynx, or the larynx. It's starting out at the trachea. Okay? So here's the trachea. It's coming down. The 2 branches here are the primary bronchi, right and left. We've got 3 lobes of the right lung, 2 lobes of the left lung. So there's 1, 2, 3 secondary bronchi to the right lung. 1, 2. Just the number of lobes. There are fewer on the left side than the right side. Right. Yeah. Yeah. And then you can see these little light blue structures within each lung. Those would be, or within each lobe of the lung, those are the tertiary bronchi. So this lobe would be broken down into what are called bronchopulmonary segments. They're not visible here as distinct, but those would be the tertiary bronchi. And you don't see anything smaller than that on this model.
So those little bunches of grapes are microscopic.
Sure, for sure, yeah, yeah, okay. So here is this structure here is the trachea. It divides into right and left primary bronchi. There's 3 secondary bronchi to the right lung, 2 to the left, and then within each lobe there are these light blue structures. Those are the tertiary bronchi. You don't see anything smaller than that on here. Did you guys want me to do it again, or did you?
I got
Trachea is here, right and left primary bronchus. 3 secondary bronchi on the right, 2 on the left, and then these small structures are tertiary bronchi. Okay. Trachea is here, right and left, primary bronchi, 3 secondary bronchi on the right, 2 on the left. The light blue ones are tertiary bronchi to bronchopulmonary segments.
Is this fairly to scale?
No,
No.
this is huge.
Well, I mean,
Oh, to scale. In proportion?
Not
I'm not sure.
I mean, the size of a lung, is it like a camelback size, or is it smaller than a camelback?
There's different sizes on your back.
The water you wear on your back. No, lungs are small.
I mean, are they like this big?
No.
Are
I mean,
they this big?
so my heart would be about the size of
Right.
my fist. So my lungs would be a little bit bigger than that, but not a whole lot bigger than that.
Okay.
I mean, they have to fit within about this space.
I mean, I've cut up a deer before, and their lungs are quite long. So,
Yeah.
but I'm no anatomy person, so I'm asking, like,
I've
They're bigger, but they have to run away from you.
They don't even run.
I've never cut into a deer to see the size of a deer lung in comparison to a human lung,
which you've cut into before.
which I have cut into before.
Okay.
So my rib cage goes down to about here,
Sure.
and so my lungs are within
That's not-
within there,
Gotcha. Well, sure.
right?
Right.
Yeah. So you'd have to be a pretty tall person to have lungs the size of a camelback water pack.
That's about the size of a deer lung, though. That's
Right. No. So our lungs are not that big.
okay.
Yeah. Yeah. So anyways, trachea is here, primary bronchi here, 3 secondary bronchi to the right lung, 2 to the left lung. The light blue are the tertiary bronchi. Good with that? Yeah. Did you guys see those okay? Okay. Okay. With these structures on your handout, I want you to put a star next to the pharynx. Because in a little bit, we're going to talk about structure-function relationships in these conducting zone structures. And we're going to see that the structure of the pharynx varies a little bit because it's a common passageway for air and food and liquid. And so when we talk about some of the structural features of the larynx, there's going to be some modifications there to account for it serving as a dual role as a passageway for air and for food and water. Okay? Are we good with these structures? Okay. We're going to take a little digression away from looking at structures to talk about how air gets into the lungs. Mammals use a negative pressure system to inhale, so to suck air into the lungs. And so we're going to talk a little bit about this negative pressure system. And in order to do so, we have to have an understanding of Boyle's law. Okay? So I have a YouTube clip that does a really nice, I found one, that does a really nice job of explaining Boyle's law.
In order to understand the
Sorry about that.
mechanisms of breathing, it's important to understand the relationship between pressure and volume. This is explained through Boyle's law, which states that the pressure of a gas in a closed container is inversely proportional to the volume of the container. What do we mean by this? If the size of the closed container is decreased, the pressure of the gas inside the container increases. And that if the size of the container increases, the pressure of the gas inside the container decreases. Imagine a gas, shown here as blue molecules, inside a cylinder with a movable piston and a pressure gauge. If the piston is pushed down, the available space inside the cylinder is reduced, which compresses the gas. This means that the same number of gas molecules have less space to move around in, with the effect that they strike the walls of the cylinder with more frequency. If you observe the pressure gauge, you'll see that the pressure doubles as the volume is reduced to half. Conversely, if the piston is pulled up, the available space inside the cylinder is increased. This means that the gas molecules have more space to move around in, with the effect that they strike the walls of the cylinder with less frequency. In this case, as the volume doubles, the pressure is reduced to half. And it's these differences in pressure caused by changes in lung volume that allow us to breathe.
Okay. So when we talk about ventilation, we have to keep in mind Boyle's law and this inverse relationship between volume and pressure. And when I cover this topic with my students, I emphasize to them that the way it works in the lungs is that we change the volume so that the pressure changes. Okay? And I emphasize this, we talk about it, but inevitably when I give the exam, someone writes, the pressure changes, so the volume changes. The volume changes, and by changing the volume, we change the pressure. Okay? So I want to show you this model first, and then I'll explain the physiology behind how we change the volume of our thoracic cavity to change the pressure. This is a little jar. It has a stopper in the top that has a hole bored through it, so this is the way that the air is going to get in. The two balloons are representing the two lungs, and this membrane on the bottom is representing the diaphragm. Okay? If I contract the diaphragm, I increase the volume inside, and you can see subtly what's happening to the balloons. It's pretty subtle, but you should see some air going into those balloons. You see them inflating a little bit when I contract the diaphragm? Okay. What's happening is by increasing the size in here, I've dropped the pressure below the pressure in this room, and the air flows in. Okay? So the two pressures that we are concerned with to make this happen is atmospheric pressure, the pressure in the atmosphere, and alveolar pressure, the pressure inside of the alveoli in the lungs. And in order to inhale, we have to make the alveolar pressure lower than atmospheric pressure, right? Atmospheric pressure isn't going to change while we are, for example, in this room. Let me do this. Okay. So at sea level, PATM, or atmospheric pressure, is equal to about 760 millimeters of mercury. Okay? And we need to get the pressure inside of the alveoli to be just lower than that. We don't have to make dramatic changes. Mm-hmm. About atmospheric pressure versus alveolar pressure. Sure. So atmospheric pressure. PATM is about 760 millimeters of mercury at sea level. Okay. As you rise to higher altitudes, the atmospheric pressure drops. You're welcome.
Measured in millimeters of
Mercury? It's just the unit of measurement for pressure. Yeah. So we need to get alveolar pressure just below atmospheric pressure for inhalation to happen. The way we need to do this, the way we need to drop the pressure is by increasing the volume of the thoracic cavity. Okay? So we need to increase volume to decrease the alveolar pressure. Okay? The way that we do that is by contracting muscles. There's two groups of muscles that contract to increase the volume of the thoracic cavity. The first are called the external intercostals, and these are muscles that are between your ribs. I'll write the name on the side for you. And the second muscle that contracts is the diaphragm. Okay? So your external intercostal muscles contract, and so does your diaphragm. What I generally have my students do is stop writing and just put their hands on their thoracic cavity and just feel what's happening as you inhale and exhale. What do you feel? Anything? You feel muscles changing, right? What is it doing to your rib cage? It's expanding, right? So you feel a rise and then a fall of the rib cage. The rise is created by contractions in these external intercostal muscles. And if you think of your ribs like a bucket handle, as those external intercostal muscles are contracting, the bucket handle is swinging outwards and upwards. Your rib cage is swinging out, is moving out and up. That's increasing the size of the thoracic cavity. Also, your diaphragm is this dome-shaped muscle. When it contracts, it flattens. That also increases the volume of the thoracic cavity. I don't believe you can feel that when you put your hands over your thorax. Okay? So by these two muscles contracting, the volume of the thoracic cavity increases. And with an increase in volume, we have a drop in pressure. Okay? Pressure drops, and air moves from the outside into your lungs. Okay? So that's an active process. When you exhale, exhalation under normal quiet conditions is a passive process. These muscles that contracted with inhalation are going to relax. The ribs will move down and in. The diaphragm regains that dome shape. We get a drop in volume, and that increases the pressure. Okay, so with exhalation, these muscles relax, and the pressure inside of the alveoli will rise to 761, 762 millimeters of mercury. It doesn't have to be dramatic. Just subtle changes in pressure are enough to allow us to ventilate air. Okay? Yeah?
What's the approximate measurement for inhalation for pressure?
So this is atmospheric pressure. This is the pressure in the alveoli that allows air to come into the lungs.
So it
Drops to 700, right, from 760 to 758. Air comes in, the atmosphere, the pressure in the atmosphere and the pressure inside your alveoli equilibrates, right? And then you need to increase that pressure to exhale. Other questions? Yeah.
Just what? I've often been told that snorkels are only so long because if you go down under the surface of the water, you do not have enough pressure. You cannot expand your lung far enough to pull the air into it. I think the longest snorkel I've ever seen is like 18 inches. So when you see these Hollywood movies with bamboo, you know, you think it's
I wish I knew the answer to the question that you're asking. I know there is some changes in pressure when you go down further, and I know that impacts the oxygen, but I don't know the answer to that question. Yeah?
If I may,
Sure, absolutely. We're all here to learn.
it can't be longer than the volume of your lungs, because you need to be able to expel enough bad air to allow
Sure.
new air to come in.
Yeah,
right? So
that makes sense.
limited by
Yeah.
the volume of your lungs.
The volume that you normally inhale and exhale with a breath. Yeah, that makes a lot of sense. Yeah, thank you. Are we okay with this idea? Yeah? Things get a little different when we talk about forced inhalation and forced exhalation. There are different muscles that get utilized for forced inhalation and forced exhalation, but for our purposes today, we'll just stick to quiet breathing. Okay? If you want to know more about forced inhalation and forced exhalation, I'd be happy to share afterwards. Okay. Oh. I did something.
We actually tried that as a kid, going down in a bowl with a hose. And when you get down a little bit,
Yeah.
there's too much water pressure. You can't expand your lungs.
Aha.
So, yeah, because we had the great idea of just hang out in the bottom with a hose. And
Oh.
you got down 2 feet, so you
Couldn't do it.
couldn't even breathe in. It just wouldn't work.
Huh. I would have never thought to try that experiment.
It was a stellar idea until we tried to put it into practice. So we almost drowned.
I'd like to hear the stellar idea. I can't hear.
He tried to go down to the bottom of the pool using a hose to breathe through.
doesn't work.
Didn't work.
No, I have kids that try it.
Ah. Okay.
Fortunately, they were all lifeguards, and so we had plenty of people on hand to pick their butts off the water.
Okay. A few more items that I want to talk about. Some structure-function relationships in the airways. So we just talked about how the air gets into the lungs based on changes in volume to create changes in pressure. But the way that the airways are designed enhances not only the ability to get air into the lungs, but the ability for us to get clean air into the lungs. And so there are numerous structures along the way that filter the air. So remove dust particles, pollen, and other foreign particles from the air before it gets into the lungs. And so I have a space for you in number 2 to fill in the blanks with some of the structures that filter the air, and also to put in the epithelial tissue type that these structures are lined with in order to filter the air. So I'm going to write the name of the tissue type down, and then we'll talk about where it's found along these airways. I have a picture. Oops. How did that jump? Huh. Huh. Must be on a timer that I didn't put it on. There you go. Okay. Well, if it moves again, I'll have to put it back. This is the tissue type that is found in the airway structures that works to filter the air. It is called, and I will write this on the board after I say it is called pseudostratified ciliated columnar epithelial tissue with goblet cells.
I don't have enough space for that.
It just jumped again.
Pseudo.
We're going to just do this.
Pseudo with sex.
Pseudostratified. Ciliated. Columnar. Epithelial tissue. One, two, three, four, 5 words. Pseudostratified, ciliated, columnar, epithelial tissue with goblet cells. So it's 1, 2, 3, 4, 5, 6, 7, 8 words.
You could win at Scrabble with that word.
Now, if you look at the picture, and I'm going to try to put this back into presentation mode. I'm not really sure what's happening here. The name makes some sense as far as naming epithelial tissues go. The cells look, they all touch the basement membrane, but they look like they're in different layers. So instead of being stratified, it's pseudostratified. Okay? Ciliated, there are cilia on the surfaces of all of the cells. Columnar, they look like columns. Epithelial tissue, it's an epithelial tissue type, and dispersed within the columnar cells are these mucus-producing cells called goblet cells. Okay, so the name is not perhaps as intimidating as it first may seem. In a 50-minute period, I say pseudostratified ciliated columnar epithelial tissue with goblet cells about 10 or 15 times, and I trip over my tongue as I do it.
You haven't made an acronym for this yet?
No. 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 Part of the problem with an acronym is I would want my students to write it out on an exam and not give me the acronym back.
Right.
So, yeah. So generally speaking, if I want them to write something out in its full, I don't use an abbreviation in class because that teaches them to use the abbreviation instead of the full. So, as painful as it may seem, pseudostratified ciliated columnar epithelial tissue with goblet cells is what we go with. Yeah. So the goblet cells produce mucus. The mucus traps dust, pollen, and other foreign particles. And then the cilia sweeps that mucus-trapped dust away from the lungs. And generally speaking, you swallow it. Okay? Yeah?
Is this where people that have asthma get into problems?
It's one place where people with asthma can get into problems. Right, that's what we're going to do next, is where you find the pseudostratified ciliated columnar epithelial tissue with goblet cells. So your blanks are where it's found that we're going to fill in. So structures that help to filter the air, so structures that have this epithelial tissue, the nasal cavity does. The nasopharynx does. Yeah, I'm not really sure what's happening. I'm going to have to try to fix that during the break. That didn't happen yesterday. So remember I put a star beside pharynx before and I told you that that was a common passageway for both air and food. Your pharynx is divided into 3 regions. The region that is just behind your nose is your nasopharynx. The region just behind your mouth is the oropharynx. And then the connection between the pharynx and the larynx is called the laryngopharynx. The nasopharynx is the only part of the pharynx that has pseudostratified ciliated columnar epithelial tissue with goblet cells. We're just going to do that. Okay. This is the lining of these structures, and that provides the primary filtering. When you get deeper into the bronchioles, there are some other cells that act as macrophages to catch anything that hasn't been previously caught by this epithelial tissue lining. But it's primarily this epithelial tissue lining that traps. Okay? So in your first box, you can write, or in the first line, you can write nasal cavity. The second line, you can write nasopharynx. The third line, you can write larynx, L-A-R-Y-N-X. The fourth line, God bless you, trachea. And the fifth line, you can write bronchi. And this is meant to include the primary, the secondary, and the tertiary. They all are lined with this epithelial tissue. Okay? The oropharynx and the laryngopharynx are lined with a non-keratinized stratified squamous epithelial tissue, not important for us, but that is a stronger epithelial tissue to resist the abrasions of the food that you're swallowing. Okay. Yeah. The oropharynx and the laryngopharynx have a different epithelial tissue type that is able to resist the abrasions when you swallow food.
Does it have just as cool the name?
It's called non-keratinized. Stratified squamous epithelial tissue. So squamous cells are flattened. Stratified means that they're in layers. Okay? And non-keratinized because they don't produce the protein keratin, whereas the stratified squamous epithelial tissue in your skin does.
And that's in your mouth.
This is in your mouth. Well, it's in your,
Behind your mouth.
right, as you're swallowing the food, the lining is non-keratinized stratified squamous epithelial tissue. Okay? Are we good with that? Okay. Okay. The next item that I want to talk about is it's very important for these airway structures to be held open so that the air can successfully pass from the external environment down into your lungs. So the airways have to be kept patent. Patent meaning open and unobstructed. P-A-T-E-N-T. So if you flip to the top of page 3, I have a bullet that says structures that keep the airway patent. Okay? And as the air makes its way into the lungs, it passes through the structures that we previously mentioned, and those have different anatomical features that help to keep them open. Yeah?
Is there a sixth?
There was not a sixth. No. There was a sixth blank, but not a sixth structure. Yeah, sorry about that. I'm sorry?
Nose hair.
Nose hair. Yeah. The structures that keep the airways patent. Starting with your nose, you have a bony framework and a cartilaginous framework. So you can write nose, bone, and hyaline cartilage. So there's different cartilage types. There's fibrocartilage, there's elastic cartilage, there's hyaline cartilage. The type that you find in your respiratory system structures is hyaline cartilage.
What is the definition of patent again?
Patent, open and unobstructed. Yesterday morning, there was an accident on the Pennsylvania Turnpike. It was not patent. It was closed and obstructed.
Where was it? I didn't pass it.
It was coming down from the Northeast Extension. It was between Quakertown and Lansdale.
Missed that.
Yeah. So I had to go around. When an airway is not patent, there's no option to go around.
Poke a hole.
That creates other problems. Okay. The next structure, the pharynx, has in its wall skeletal muscle, which gives it some structure and helps keep that airway open and unobstructed. Your larynx has hyaline cartilage. And I'm afraid to put this back in PowerPoint mode, but what you would be able to see on this figure are the different cartilages of the larynx. The trachea has incomplete rings of cartilage, so they're C-shaped cartilage. Okay. And hyaline cartilage, it's a type of cartilage. So you have elastic cartilage, you have fibrocartilage, and you have hyaline cartilage. The type that's found in the respiratory system is the hyaline cartilage. Hyaline cartilage is also the cartilage found on the ends of your long bones at your joints. It's also found in growth plates in growing kids. That's the cartilage type. Okay. So what I traced here is the incomplete ring of cartilage found in the trachea. Okay. As we move further down the airways, we go from these incomplete rings to plates. So when we get to the bronchi, the primary bronchi start out with these incomplete rings. Those incomplete rings will become smaller and smaller and smaller and eventually be plates of cartilage. Again, it's hyaline cartilage. And then by the time we get to bronchioles, the cartilage is gone and it's replaced, I'll put it up top, but just know that this is last. The bronchioles do not have cartilage, instead they are smooth muscle in the walls. Okay. So the bronchioles do not have any cartilage anymore. As the cartilage decreases, the walls are replaced by smooth muscle. Okay, so the bronchioles have smooth muscle instead. Okay. This smooth muscle in the bronchioles provides them an opportunity to function as a regulator of airflow. So structures that allow for alteration to airflow are the bronchioles. And when we talk about flow, in order to understand how the bronchioles alter flow, we need an equation. I'll just let that go. Flow is directly proportional to delta P, which is our pressure gradient. We already talked about that a little bit with making sure that alveolar pressure is less than atmospheric pressure to bring air in, and that alveolar pressure is greater than atmospheric pressure to bring air out. So we talked about the pressure gradient, delta P. And there's an inverse relationship between flow and big R, which is resistance. Okay. When we talk about resistance, big R, it's equal to pi times mu, which in this case is viscosity, pi times viscosity times L, which is length, divided by small r raised to the fourth, and this small r is radius. Okay. radius of the structure through which flow is happening. So for today's purposes, we're talking about airways. The next time that we meet, we're going to be talking about blood vessels. We're going to use these same equations. For airways and for airflow, this is the term that we're really interested in. If we increase the radius, we're going to decrease resistance. Right? If we decrease radius, we will increase resistance. Yeah. Pi as in 3.14. No, this is pi. Yeah. Sorry about that. So when we talk about bronchial diameter, if we want to increase airflow, we would increase the diameter of the bronchiole because that would decrease resistance. Okay, if the bronchiole diameter decreases, that will increase resistance and it'll decrease airflow. And what I've done, I found a little exercise from Science Buddies. I took 2 cups, I made holes in the cups, and I put 2 different diameter straws through to model what changing the diameter of the tube does to flow rate. I just took some water and used some yellow food coloring so that you can see it. If you don't want to use water, you can use sand. If you don't want to use cups, you could use funnels that have different diameter openings. And I'm going to pour it simultaneously, and then I'll flip it around so you can see. Which cup is emptying faster? This has the larger diameter straw. This has the smaller diameter straw. Right? So you can see the difference in flow rate there. If you have a group of students and you don't want to give them the equation because that's too scary, you can just focus on how changing the radius changes airflow.
So what is the mu value for this non-character?
It would be the viscosity of the air.
Oh, okay.
So generally speaking, you're not going to change the viscosity of the air, and you're not going to change the length of your airways. When we talk about blood and blood flow later in the week, if you have a situation of dehydration, that would change the viscosity considerably. If you have someone who has gained a lot of weight in a short period of time, you're going to have longer vessels, which is going to impact the resistance to flow of blood. And so those other factors are going to be much more important for us to discuss when we talk about blood flow. But nevertheless, they're part of the resistance equation, and so I wanted to give you the full equation. Does that make sense? Yeah? Okay. We'll be good with that. Okay. Finally, the structure that allows for gas exchange is the alveolus. Ugh. Just going to leave it that way. The alveolus is this cup-shaped pouch here. Okay. Notice how it is in very close proximity to a number of capillaries, pulmonary capillaries, and there are lots of alveoli in the lungs. So we've maximized surface area because there are a lot of them, and the diffusion distance is minimized because of the proximity of the pulmonary capillary to the alveolus and the thinness of these structures. So the alveolus is only a single cell layer thick. The tissue that makes up an alveolus is called simple squamous epithelial tissue. And this is found in the type 1 alveolar cells, which are the cells that form the walls of the alveolus. Just below this epithelium is a small, what's called basement membrane. All epithelial tissue sits on a basement membrane. There is a small or thin basement membrane for the pulmonary capillary. And then the pulmonary capillary also has a simple squamous epithelial tissue layer. So these 4 layers are sandwiched together to form what's called the respiratory membrane. And if you want an order of magnitude here, we're talking less than a micron thick or less than a micron thin. Okay, so here is the respiratory membrane. Here's the wall of the alveolus. This pale blue here is the epithelial basement membrane, and then here is the simple squamous epithelium of the pulmonary capillary. And I left you some space, God bless you, if you want to draw your own respiratory membrane, or maybe write sort of the order of the layers that the gases would pass through to get from the alveolus into the blood. Okay. Do you guys have any questions? No? What I thought might be a fun way to end, I took a beach ball and I put questions in each of the panels. And if we toss the ball around, whatever panel your thumb lands on, your right thumb, you answer one of the questions from the panel. There's 3 questions, so you have three to choose from, and you can pick whichever one you want, and then toss it to someone else. So we can all end having a ball. Ready? You have a question? You want the ball? Go ahead. There you go. You can start. So wherever your thumb is, pick a question. Well, you're grabbed with your left, so you still can answer.
Okay. What is the respiratory surface for fish?
Yep. For fish. The respiratory surface for fish.
Uh-oh. Look through my notes here. All right, let me tackle the second one.
Yeah.
Boyle's law states an inverse relationship between pressure and volume.
Yep. Very good. You can toss it to someone else.
Who's got it?
You said we had to catch it, right?
Well,
Let's see here. Inhalation happens when the atmospheric pressure is greater than the air pressure.
yep. Very good.
I try to get it too.
Lumen of the bronchioles is encircled by smooth muscle.
Yep. The lumen of the bronchioles is encircled by smooth muscle.